Welcome. In this video, I'll explain how to perform spectral unmixing using the MILabs Vector 5 system here, which is available as a CT optical spec pad system or also as a modular system with some parts of these. The software is called MILabs Preclinical, and you can find more information in this paper here in Theranostics or on our website. So in this example, we imaged several tubes with brake light fluid inside. Here pink, yellow and turquoise, and also red and orange. But we will focus on these here in the upper part. And these brake light tubes were imaged in a holder for 96 well plates here. So let me show you the data set here in the software. So we acquired 4 channels, and we can go through the channels here. And with pressing F5, I can automatically set the windowing. So let's go back. So this is the first channel, and here the turquoise tubes are really bright. In the second channel, everything is bright, but the yellowish tubes are brighter than the others. The third channel, the pink or the reddish and orange ones are bright, and in the fourth channel, also the reddish tubes are bright. Okay, let's go back to the PowerPoint. So this is an example for bioluminescence imaging. Well, it's more like chemoluminescence, and we use these brake light tubes here, and the 4 channels we used have the wavelengths 534 nanometers around here, like in the green range. And then the other 3 channels are here in the reddish domain. Here, so the channels are not really optimal for these brake light tubes, but the system was also not meant for the brake light tubes. But it's still a nice example. The channel width is around 40 nanometers for each channel. So now that we have this multi-channel image here, we can set regions here by drawing a region, for example, and adding a class turquoise. Then with F3 and F4 we can segment a region here. We can do this for all the channels, for all the tubes, the different colors. But let me load an existing segmentation here where pink, yellow and turquoise are segmented. And now here with the menu item unmixing, we can save the class spectra of these. That results in basically an Excel file, and I've prepared it here and there. It's a comma separated file. You can open it with Excel and then look at the spectra here, and you can see the turquoise is very strong on the lowest channel. The yellow tube shines in the lower 2 channels, and the pink tube shines in the higher channels, but it's all pretty much mixed up. And this is another visualization of this data, and you can see that the channels don't really separate the tubes nicely. Well, maybe the last channel here is very strong on the pink tube, but there's no channel, for example, that really separates the yellow tube from the other channels. So, but what we can do is now to perform spectral unmixing here, because the 3 spectra are all different. So what we will do is every pixel can be decomposed into these 3 spectra. So every pixel has its own spectrum, and there will be coefficients computed that explain the spectrum of the pixel with these 3 basis functions, or these 3 spectra. And this is performed here using spectral unmixing. Let's start this, and you have to select the spectra file that was created previously, and now it's running in the background and computing. And let me explain what it does here. So it's cast as an optimization problem here, and these are the three known spectra here: pink, yellow, and turquoise. They are vectors, four-dimensional vectors, and these are the weights of the spectra, and they are being computed: alpha, beta, and gamma. And this sum, this weighted sum, is then our predicted spectrum, and this is the measured spectrum of the pixel. And then the squared residuum is computed, and this function, this term, is being minimized with respect to the three weights of the spectra: alpha, beta, and gamma. This minimization is performed for each voxel, or a pixel in this case. It's a least squares problem, but we are avoiding negative values here because there's no negative concentration in reality. And this is performed using multiplicative updates, and the optimization is done using GPU-accelerated gradient computations, as shown in this paper here. And this is an iterative approach, and around 1,000 iterations are needed. So let's see if it's done by now. Yes, it's done. And now it's loading the unmixed results as underlay here. This is a file with 3 channels now. We can go through these channels. So the first one is this turquoise one. Here the second is for the yellow ones, and the third is for the reddish ones. So they all these 3 reddish types appear in one, appear bright in this channel. Now we can save this image here. I've saved it already, and you can load the previous segmentation that we used, and we can use a quantification here using statistics, class statistics underlay, save the quantification of these regions and the channels as Excel files, and then look at them. So let's prepare this in the PowerPoint file here. So in the unmixed quantification, it's nicely separated. So here in the pink channel, only the pink tube is bright, in the yellow channel only the yellow tube, and then the turquoise channel only the turquoise tube. We can also load this image now as overlay here, just to have a look at it. This is now shown with false colors, and you can see that they are nicely separated, nicely clean colors. Okay, that's it. Thank you.